So today let's talk about biogenic studies. Why we are studying this biogenic studies? What are these biogenic studies? If we are studying this biology and genetics combinedly, that is called as biogenic studies. It will study how the primary and secondary metabolites are synthesized in the living cells. Example, we have seen shikimic acid pathway. Now we know this pathway, but previously how we have introduced this pathway with the help of this biogenic studies only. It have explained how to find it out. First thing, how to extract this one, and what are the experiments needed to extract this isotopes. So all this comes under this biogenic studies only. For example, if I'm studying primary pathway and secondary pathway, but how I have recognized that primary or secondary pathway, from where I have taken that, and how I recognized those are the metabolites. That's because of this biogenic studies only. Okay. Radioactive isotopes. Radioactive isotopes. So what are these radioactive isotopes and why we will use this, and why do we need this radioactive isotopes in plants, from plants? Okay, so before going into this radioactive isotopes, let's see what are isotopes. If any one or two more species of atoms of a chemical element with the same atomic number and position in the periodic table and nearly identical chemical behavior but with different atomic masses and physical properties, those are called as isotopes. For example, if I have carbon 6, the atomic number will be 6 every time. Whenever you write this, you will be having same atomic number 6, but the atomic masses will be differing. For example, if you see all these carbons, you have same atomic number but different atomic masses. So C6 12, C6 13. Here what is happening? You will be having same number of protons but different number of neutrons. Actually, in this carbon you will be having protons and neutrons. In this, if you have 6 protons, then you will be having 6 neutrons. Okay, that's why you are getting 12. 6 plus 6, 12, right? And if you see in this one, you will be having 6 protons but 7 neutrons. Okay, then it will become 30. So these protons will be same in, if it is C14, C614, then you will be having 6 plus 8, 14. Okay, in every carbon atom 6 will be same. That means protons will be same but different neutrons. These are isotopes. So let's see another example of these isotopes. So let's take hydrogen. Hydrogen atomic number will be always 1 in periodic table, but the atomic masses can be 1, 2, 3. The protons will be 1 only. If you see here, you will be having 1 proton and there won't be any neutrons in first one. That's why you will get 1 only. But in second one, you will be having 1 proton plus 1 neutron, which is 2. 2 atomic masses. Third one, 1 proton plus 2 neutrons, 3, so on. So in this isotopes, you have two different types of isotopes. One is radioactive isotope and the other one is stable isotope. So first, let's talk about this radioactive isotope. So this radioactive isotopes is also known as unstable isotope. Why it is called as unstable isotope? So if you have these isotopes like carbon and hydrogen, the nucleus you will be having neutrons and protons. When you have so many neutrons and protons, there will be excess of energy. Due to this high energy in this nucleus, this energy will start to, they won't be able to stay stable in these atoms. So they want to come outside. But they will come outside, they will be released in the form of radiations. How? They will bombard with each other and they will emit these radiations. These radiations will be in alpha, beta, and gamma forms. So when these radiations are coming out, that is called as radioactive isotopes, as they are, why we are calling as unstable, because this energy is unstable inside that one, so it wants to come out. There is so much of energy in that atom which this atom is unable to bear it, so it will release this energy. When it is releasing, it will come out as a radiations. For example, we have few radioactive isotopes which will emit these radiations, and let's see what are the uses of that. For example, H13 and C614 and S35 and I131. So these are the isotopes which are known as unstable isotopes. These are used for X-rays and ultrasounds. So we use this, that's the reason X-rays and ultrasounds are nothing but radiations only, isn't it? That radiations are due to these isotopes. So that is the use of this radioactive isotopes. Okay, and next let's see the stable one. The stable, when there is after releasing all this energy outside, the isotopes will become stable. Okay, so those isotopes, they don't emit any radiation because they is stable, so there won't be any emission of radiations. Those are H2, C13 and N15. Okay, so for every radioactive isotope we have medicinal uses. For example, iodine 131, which will like use as a X-ray to locate this brain tumors, monitor cardiac rhythms, and liver to see, monitor liver and thyroid activity. Okay, so next let's see this plant biosynthesis. What is this plant biosynthesis? So traditionally, when we have recognized the medicinal uses of these plants and everything, they want to know how we are getting that medicinal uses. Okay, from plant, for example, if I am taking any herb, Hemidesmus indicus or anything else. So all the scientists have wonder how we are getting those metabolites, how we are getting this secondary metabolites or primary metabolites. So after knowing this biochemist, they started processing all the primary metabolites, and organic chemist want to study on this secondary metabolites. Okay, to study this primary metabolites and secondary metabolites, we have four major techniques used in this biogenetic pathways. We have four major techniques. First one is trace technique, and next one is use of isolated organs and tissues, and third one is grafting method, and fourth one is use of mutant strains. So let's see what is this trace technique. So when one or more atoms of any expected precursors of target plant constituents replaced by isotopes, and used as markers in biogenic experiment, those are called as this tracer techniques. So this tracer techniques can be defined as when we are utilizing any radioactive compounds to find out the intermediate compounds or intermediate metabolites or primary metabolites or any secondary metabolites in a plant, then at a certain rate and a time, then that is called as tracer technique. So the examples of this are C14 and H3. So when we are selecting this radioactive isotopes, we have to remember the half-life of that radioactive isotope. That means when we are using this, the half-life should be very flexible to the patient. And next one is non-damaging nature. So when I'm using this radioactive isotope, it should not damage my body. So next one is should used or mostly used in biological researches only. So these isotopes, they have to be used in this biological research only. We should not use for any other purposes. So let's see the methods used for this recognizing this radioactive isotopes. So before selecting these methods, for radio labeled compounds, we have to consider few steps. That is, when we are introducing this radioactive compounds into the plants, we should select a particular place. For example, if we want to do on root, we have to select the root only. If we want to do whole plant like from roots, leaves and stem, we have to select that kind of method. Okay, and next one is the time of introducing of this radioactive isotopes should be correct, so that the labeled precursors should be available during the biosynthesis of the compound under investigation. When we are doing this investigations of this biosynthesis, that should be correct. Okay, for example, if I'm introducing the radioactive isotopes now to study some secondary metabolites, so I need for secondary metabolites I need only 2 hours of time. So that till the 2 hours it should be intact with that plant. So the time of administration should be correct. And next one is we have to give minimum dosages of this radioactive isotopes. Because why minimum? To avoid the involvement of this radioactive isotope compounds with other biosynthetic reactions. So let's see the different methods of this in different methods of introducing this radioactive isotopes into the plants. So let's see the methods. First, root feeding, stem feeding, injection method, wick feeding, floating methods, spray method. So let's see one by one. The first one is root feeding. So root feeding is, for root feeding, we need this secondary metabolites or primary metabolites from the roots. So we will fill this one, like we will take a beaker, we will fill the radioactive isotopes and we will insert this plant, but till roots only, because we need only roots, right? So till roots we will insert this plant. We have to keep for some time till this radioactive isotopes absorbs into this roots. Then we have to remove the plant from this isotopes. The examples of root feeding are tobacco alkalides, and Datura alkalides. Next one is stem feeding. So this method we need for this radioactive isotopes to be in this stem, so that we can recognize what are the important activities in that stem. Okay, so we will cut this stem into half. So after cutting, we will leave that into this beaker which contains radioactive isotopes along with nutrients. Direct injections. So this is the most easiest introduction of this radioactive isotopes, because it is very easy. We have to take the radioactive isotopes and we will inject that into the stem. The examples of this direct injections are ampliferous and opium. Next one is floating method. This floating method is we use this technique
Like we will take small amounts of material. We will take any, if we want any leaf or if we want any flowers, we will take that and we will insert that into, like we will take a beaker of this radioactive isotopes along with nutrients, and we will cut this leaves or flowers, and we will let them to float on this radioactive isotopes. Okay, that is radioactive isotopes. We use this method when we have very less isotopes. That means we have very less quantity of this isotopes. Spray techniques. This spray technique is wherever we need. If we want in stem, we will spray these isotopes near the stem. And if we want on leaf, we will take the specific specific leaf, and we will spray onto that leaf, and after spraying then we will test the pathways. The examples of this spray technique is steroids. So how we will do this spray technique? First we have to mix this radioactive isotopes with water, and we have to spread all over the leaf, and after spreading, this compound will be absorbed by the leaf. After some time, we have to do the experiment. And last one is wick feeding. So if wick feeding is, if you see this diagram here, this we will be attaching one wick. Wick is nothing but a cotton thread. So we will attach this wick with pots. Okay. So we will insert this wick into radioactive isotope bowl, which so if you see here, this is a radioactive isotope bowl in which you have radioactive isotopes. So we are inserting the wick into this. So this wick will be observing this radioactive isotopes into this pot. Okay, so it will grow it will absorb the radioactive isotopes, and finally you are left with no solution. So after introducing this radioactive isotopes into the stem or plants or anything else, we have to detect that radioactive isotopes, right? To detect that one, we have 8 types of detectors. First one is Geiger-Müller counter, and next one is auto radiography, and scintillator counter, liquid scintillator media, gas ionization chamber, Bernstein Bellantine counter, mass spectroscopy, NMR spectroscopy. So first let's see this Geiger-Müller counter.
So the first one is Geiger-Müller counter. So if you see this equipment, you have, this is the main Geiger-Müller drum, and this is amplifier, and you will be having digital counter. Let's talk about the background story of this Geiger-Müller counter. Giger is a scientist. He is a professor who have introduced this equipment, but it haven't finished completely. So he left that in middle way, and he died. And after few years, his student, who is called as Muller, he have completed this experiment for his professor, and he have introduced this counter as Geiger-Müller counter. Geiger is his professor and Muller is the student. So let's see how this work. So you will be having this tungsten in middle, which is a anode. So this is anode, and to the lining of this you will be having cathode. So when any radiation passes into this, what will happen, this will get, so when there is when we are introducing any radiation into this, it will ionize the gases inside it. So you have so many gases inside this, so they will get ionized, and after they get ionized, the ions in this, ions and the electrodes inside this, ions and electrons inside this, they will get attracted towards this electrode, and this will produce the electric current. Okay, and this electric current is will enter through this electrode, and it will pass through this one, and it will go into the amplifier. From this amplifier, that electric currents will go inside this and it will show the reading. This is how we will recognize the radiations. Okay, and we have this not only in plants, we have this gigamuller counter everywhere. Like we used to have this small radios. Okay, so when we have these radios and we are using our phones beside that radios, it will give a type of sound. That sound is due to this gigamuller counter only. So this gigamuller counter started to recognize the radiations which are coming out of this phone. Okay, that's why it will make that sound. So let's see the advantages of this gigamuller counter. This is inexpensive and this is very small equipment, so we can carry anywhere. Like we can keep it in a bag and we can carry it, and you will be having longer duration. As this is tough equipment, it will be having long duration, and it will detect all types of radiations. So let's see the disadvantages too. So we can collect any type of radiations like beta, gamma and alpha, but we can't differentiate this what type of radiation. Okay, whether it is alpha or beta, we won't be able to recognize this one. Okay, so next let's see the scintillator. So this is the scintillator. So the scintillator consists of this first one is this is the main scintillator, and this is photo multiplier tube, and you have this connectors. So when this radiations enter into this, inside this scintillator you will be having, so as this radiation is energy, it will go into this whole scintillator and it will start to bombard to the walls. So when this low energy photons, when they bombard to this photocathode, what will happen, it will enter, it will penetrate from this one, and this will go inside. So the electrons will go inside. When they are going inside, first one electron will go and hit to this photo multiplier. This is the photo multiplier. When it is hitting, this will reflect two electrons. So if it is hitting one electron, it will reflect two electrons. Okay, when two electrons are hitting another photo multiplier, it will reflect four, because one is reflecting two means two will reflect four, right? So now it is reflecting four, and 4 will become 8, and 8 will become 16, and 16 will become 32, so on. So at the final, you will be getting a number of electrons which are collected here. This is the anode. We will be connecting to a counter here. From in this counter, we will be noting the electrons, right? So by that, by collecting those electrons, we will estimate the radiations, whether it is alpha or beta or gamma. So next one is autoradiography. This autoradiography, this is the process which we use to recognize the radioactive isotopes in biological samples, like when we are giving to any animal, or when we are giving to any human, or if we are giving to any particular cell or tissue or cell organelles, we can recognize in this one. It's almost like a x-ray. So we will take a plate like film. So this is the film, and we will keep one radiation tray here. So we will emit radiation onto this film. So when we are emitting this radiations onto this film, it will show the paths. Okay, when it is showing this paths, after some time, this will reflect in a color radiography. Okay, so we will use this to scan our body parts also. For example, if we got any hand fracture, they will keep on one slate. If you go for any x-rays, they will keep your hand on one slate. They will take this scan. That scan is nothing but they are passing some radiations onto your hand by keeping on this film. On this film, you will keep your hand. Okay, so this is your hand, and due to this radiations falling on this hand, and those are reflecting onto this film, you will be able to see the structure of this cell. So we have types of photoradiography. One is contact radiography, and the next one is liquid emulsification, and third one is radio paper chromatography. In contact radiography, this film and sample both are kept in contact with each other for a specific period of time. That's how it will give the result. And liquid emulsification, we will take a specimen. On this specimen, we will pour the gel. Like we have to keep in this line position, and we have to pour the gel on it. Paper and thin layer chromatography. In this one, we have to take a paper. So imagine this is the paper. We have to keep isotopes, and we have to dip in a solution so that it will show till how much length these isotopes have traveled. Depending on that, we will be estimating the radiations. So let's see the methods of trace studies. So in this we have three methods. One is precursor product sequence, and the next one competitive feeding and sequential analysis. So let's see the first one, which is precursor product sequence. Let's see this precursor product sequence. This is the most commonly used biogenic pathway. So we will be introducing our radioactive isotopes into this, and we will let it allow to sit for some period of time. For every isotope you will be having different time period. So after that, we will take this one, we will isolate and we will purify it, then we will confirm the sequence of these pathways. But the disadvantages of this is when we are introducing this radioactive isotope, this may not be sufficient to extract the pathways. So the end will be the common pathway. You will get the common pathway, but you won't get the specific intermediates of that, like primary metabolites or secondary metabolites or intermediate metabolites. For example, if you take shigmic acid, you will be having all this primary, secondary, and tertiary, right? But you won't get that one. You will get a common pathway, like for carbon dioxide, as I told you, like that. So that is the disadvantage of this. But when we are giving this radioactive isotopes in double dosages or triple dosages, then this will work. Then we will get the exact pathways. So, and we have learned that we should not give more radiations, more radioactive isotopes into the plants, which will damage the biogenic studies. That one also we have to keep in mind. So double dosages and triple dosages is not a problem for that. So next one will be our competitive feeding. In this competitive feeding, when we have any metabolites, the primary metabolite will be the same, and the final metabolite, which is secondary metabolite, will be the same, but the intermediate metabolites will be differing. For example, if we see any pathway, so now I am taking a pathway in which you have primary metabolite A, and you have two intermediate metabolites, but you are getting a secondary metabolite C, the same pathway. So we won't be able to recognize which is the main intermediate pathway between these two. Okay, they both will be very competitive. When that's why it's called as competitive feeding. So what we have to see, we have to go into clean studies of this one, and we have to select the proper one which has the maximum effect. For example, if A is the primary and B, it is showing, this B is showing 75 percentage of this pathway. Like if we remove this also, it is giving the same product C. Then no need of this one. And if you take this one, it is not giving a proper secondary metabolite. Then we have to cancel this one properly, and we have to select this one. So next one is sequential analysis. In this sequential analysis, we will take a plant, we will grow in carbon dioxide atmosphere, not in oxygen, but we will supply with more carbon dioxide, Okay, and then we will introduce this radioactive isotopes into this plant. Okay, then then we will take the sequence of this metabolites. I will say one example for this sequential analysis. We will take tobacco alkaloid. Okay, and we will expose this tobacco alkaloid for this carbon dioxide for 5 minutes. Okay, then we will take this roots and leaves of this tobacco, and we will insert this radioactive isotopes, and then we will see the result, we will see the pathway.
So let's see isolated organ tissue and cells. When it comes to plants, we will take these organs which are like our shoots, roots, leaves, and flower petals. They will be our organs. Okay, we will take these tissues or organs and cells, and what we will do, we will grow under a specific aseptic conditions, and after doing this we can use these in feeding experiments. For example, if I want to take anything which belongs to root, then I will take this tissue which I have collected, this organ like roots. I will take them and I will continue the experiment. I will store it aside in a aseptic condition. Okay, and when I needed it, then I will take it out and I will use that one. That is isolated organs, tissue and cell. So in this the radioactive traces will be introduced in these parts. For example, petal disc. Like when we are extracting any rose oils or essential oils, we will what we will do, we will take this rose petal disc. So if you see here, this is our disc. So this will be our petal disc. So we will take this, we will be injecting this radioactive isotopes into the petal shoot, okay, petal disc. We will get to know the active metabolite in this. For example, in rose petals, we will extract rose oil from the rose petals. And next one will be are grafting methods. When it comes to grafting methods, from small age we are seeing what is grafting and everything, and we used to do experiments. So in this grafting one, we will cut a surface of the main plant, okay, and we will take another plant which we want to attach to this main plant, okay. So the major plant for the grafting is called as stalk. So if you see here in this is called as stalk because this is the main plant, okay. So I have sliced it off. This is the stalk of datura, okay. I am taking another plant which is tomato and I am cutting this area, okay, and now I am attaching these two. That is called as grafting unit. If you see here, this is graft union. So we will wrap it with one plaster or any tape, okay, and we will let it to grow. So after some time, what will happen at this area, there will be accumulation of tropine alkaloids, okay. We can use another plants also, which is cinchona, citrus, okay. So this is grafting. Next one is mutant strain. There will be mutant strain. For example, if I am taking any plant, because of growth of fungus or any other microorganism, that will affect the plant's metabolites. Okay, though the plant belongs to same species, but due to a growth of any fungus or any microorganisms, it will change the metabolites. For example, if you see this one, this is ray, okay. In this ray you have a fungi called as claviceps purpurae. This is claviceps purpurae. So if we remove this from here, this look like this. So this is a type of fungus. It will be having legs, okay. These are legs, okay. This will go and attach it to this ray. When it is attaching the ray, if you have compared with normal ray and this one, when you are extracting any metabolites, which is very healthy ray, and when you are extracting any metabolites from this
mutant ray, what will happen? There will be different metabolites, and not only that, because of this one, the whole metabolic pathway will be greatly affected. They will the whole metabolic pathway will change because of this single fungi. So this is the end of your radioactive isotope class, ma. Thank you, everybody.